Okay, so welcome everybody to the Ataxia Global Young Investigator Initiative webinar series. As you know, the Ataxia Global Initiative is hosting this webinar series for young investigators in the ataxia field to introduce you to important aspects of various research topics. In the first webinar series of the Ataxia Global Young Investigator Initiative, each AGI working group will present an introduction into their topic. Today, Andrea, Nemeth, Stephan, Zuchner, and Matthis Synofzik will give the webinar Next Generation Sequencing Analysis in Ataxias, what it is and how it is done. Scope of the webinar is to introduce and explain the basis of exome and genome sequencing and its analysis in genetics ataxias. Focus will be on discussion of strengths and limitations of these sequencing techniques, but in particular also of their analysis. The importance of filter settings will be demonstrated by illustrative, exemplary hands-on demonstrations of how to do exome or genome analysis. The webinar will present the major international NGS ataxia pipelines and provide an outlook on the next steps in ataxia genomics.
I'm happy to introduce the speakers. Andrea Nemeth is Professor of Neurogenetics at the University Hospital in Oxford and a consultant at the University Hospital NHS Foundation Trust. Her aim is to understand the genetic causes of neurological disorders and how they can be treated. She has been involved in the identification of numerous genes associated with ataxias, intellectual disability, and other neurogenetic disorders. She runs specialist integrated neurogenetic translational clinics with a view to being clinical trials ready. She also runs a laboratory using next generation sequencing and stem cell and gene editing technologies to identify and understand the cellular basis of neurogenetic disorders. And second, Stephan Zuchner. He is a professor of human genetics and neurology at the University of Miami Miller School of Medicine. He serves as chairman of the Dr. John T. MacDonald Foundation Department of Human Genetics and is the co-director of the Hussman Institute of Human Genomics. His lab is internationally renowned for being highly successful in identifying several dozen genes for Mendelian disorders and also evaluated risk factors for complex genetic conditions. His group is amongst the pioneering groups that have promoted genome sequencing methods and advanced bioinformatics for disease gene identification, not only in human, but also in mice, worm, and Drosophila. By utilizing in-house developed software solutions, such as the widely used Genesis platform, he is currently pursuing large-scale collaborative exome and genome analysis in multiple Mendelian neurodegenerative disorders to map their complex gene architecture, including modifier genes, and finally, therapeutic applications based on genetic approaches are being developed in his group. And last but not least, Matthis Synofzik. But to be honest, I think this man needs no introduction. He's the head of the research unit Translational Genomics of Neurodegenerative Disorders and senior consultant neurologist at the Hertie Institute of Clinical Brain Research and Center of Neurology at the University Hospital in Tübingen. You definitely know him from several rare disease consortia on recessive ataxias like PREPARE, PROSPECT, and ARCA registry. He's involved in clinical research and molecular genetics, as well as molecular and digital motor biomarkers. And what shall I say? We're happy to welcome all of you, and we're especially happy to welcome you to this format. Many thanks for being here. And to all participants, I can just say, enjoy the talk, and with this I will hand over to Matthis. I think you will start.
Thanks very much, Heike and Jenny, for this really kind introduction, but also really for doing this job and hosting this webinar series. Thanks a lot. So we are going to talk, as Heike said, about NGS analysis and ataxias. What is it and how is it done? And just as a disclaimer to start off, this is going to be for starters. So if you know already about next generation sequencing, if you have done it, if you do it on the research level, you can go to bed, you can keep working, whatever you want to do, but don't keep this webinar. This will be for starters, because what we have learned is for example, I don't have a lot of knowledge about MRI or whatever, so I need to learn there. So we are all learners in some fields, and this is for learners in the S field. So it will be basic. How are we going to, what are we going to cover tonight? I will start off with a short introduction. The next point then is Sanger sequencing, panel, exome, genome sequencing. What are the differences, strengths, and weaknesses? So this is really about the sequencing platforms. And so it's a hardware, so to say, and then we go on with the software, with the filter setting. Once you have your sequencing results, what is important, how do you filter those to come to your results? And what Stephan Zuchner is going to show is that they are actually the crucial key in understanding the NGS diagnostic research reports. So if you as a clinician receive an NGS report, this is a construct which has to be influenced by many filters and many artifacts. So it's not the truth, it's not a fact, it's a construct. And this is what Stephan is going to show you. And Stephan's part will include a hands-on demo. On the fourth part, we will just present you the major international NGS Ataxia pipelines and maybe ways to contribute for you. If you have exome and genome data sets which you would like to share or want to do research upon, this will be your opportunity. And finally, then we'll have an outlook and give you some more, then really some science outlooks and what are the next steps in Ataxia genomics, and we conclude by discussions and questions. So just to start, genetic stratification. Why is genetics so important? We have all learned about genetics as a diagnostic tool. But what is really exciting in the decades to come, now in the 2020s, we are moving from genetic diagnostic to genetic theranostics, meaning we're not just doing genetics anymore to diagnose a disease, but by giving a patient a diagnosis, we are making this patient ready to trial. And it's a completely new switch. It's like really turning around what we have done in the last years. Why is that? That's because we will not treat ataxia as ataxia. Rather, we will have to genetically stratify it. And then gene A will get a different treatment than gene B, than gene C or gene D. So the treatments, the high effect size molecular treatments will be run via the gene. And this is why genetic stratification is a tool and why it's not just diagnostics, but theranostics. And just to give you some example of the druggable ataxias, we are going to see a SCAR3 trial already later this year. A SCAR1 trial is going to come probably in the next 1 to 2 years. Nemeth type C is already a treatable ataxia. COQ8A, which is ADCK3, is also already treatable ataxia. Splice mutation ataxias, meaning ataxias caused by splice mutation, there we're going to develop ASOs even on an N of 1 level. So this just shows you by doing genetics, you're just not doing diagnostics, but you're getting patients ready for trial. And for some other diseases, we do not yet have drugs on the horizon, but we make them trial ready, like RSARC, SPG7, RC1, meaning putting all the ingredients together to put them to trial. Once a compound or an ASO is available, then we can directly start with the treatment trials. So this is really a new perspective in the 2020s, and this is why we need the genetic certification. What is a problem? One of the problems, or actually one of the good things, because this means we can make the patients trial ready, is the exploding number of genes. And here you've seen in red all those genes which are associated with ataxia either as the main phenotype or as an associated phenotype. And as you can see is that the number is in particular increasing in the last, let's say 10 years. And there are almost now 200 genes which are associated with ataxia as the main or as an associated phenotype, about 90 genes which have ataxia as a main phenotype. And the number is continuously increasing. And given this high number of different ataxia genes, I think the first lesson to learn is that single gene sequencing, what has been done by Sanger sequencing, Andrea will explain you what this means, this is not really helpful anymore. We would not even start by doing one gene, because as you can see here, you can do this endlessly. You can do this for 200 genes, you will never get it done. So this means you need a new or relatively new technique, next generation sequencing, which allows you to cover several genes. Secondly, you see that the number of genes is increasing each year, new genes are being added. And this means why any hardwired NGS panel by which you enrich a certain small gene set is already out of time once you have it at place. Because if you have the results 6 or 12 months later, it will already not include the latest genes. So this is why, and this is, I think, one for the first diagnostic take home, which is I think sooner or later we get to an exome first strategy in diagnostics. And we will tell you what exome means and why exome first might be a good way to go. But what's the current status in ataxia genetics? Where are we at currently? Before coming to the next generation sequencing. Currently, we have the following field in Ataxias. In autosomal dominant Ataxias, what you can appreciate here is that about 60 to 70% of all the Ataxias are caused by SCAR1, SCAR2, SCAR3, and SCAR6. And all of them have one genetic feature in common. Namely, they are repeat expansions by a certain type of repeat CAG. Those are exonic CAG repeat expansions. So 70% of all dominant Ataxias share one mutational mechanism, namely CAG repeat scars, as opposed to single nucleotide variants or as opposed to deletions. They are caused by repeat expansions. Why is that important? That's important because CAG repeat expansions are not covered by standard next-generation sequencing. We will learn later that they might now be more and more be picked up by genome sequencing, but that's not yet part of standard analysis. Meaning, even if you do NGS, you actually miss the major part of what is causing dominant ataxia. There are other ataxias which are, however, much more rare. Those you do cover with next-generation sequencing. Those are caused by so-called conventional scars or conventional mutations. With this conventional scars, we mean all those scars which are caused by normal mutations other than repeat expansions. And about 1/3 of the dominant ataxias is still unsolved. This means we have to come up with new methods to still tackle this upper third and make them trial ready by finding genetic diagnosis. And at the very end, Stephan will tell you about new ways how we can tackle this upper part of the pie. If you look at recessive ataxias, there's one really interesting similar feature, namely the most common disease cause of recessive ataxias is again a repeat. It's not a CAG repeat, it's a different repeat, it's an internic repeat, but this doesn't matter, it's also a repeat, namely a repeat causing Friedreich ataxia. And this means also in the recessive ataxia world, the most common cause is not a conventional mutation, but a repeat expansion. And for this, you can even now add a second type of repeat. I don't have the numbers in yet because the numbers, my pie chart is a little bit older. This gene has just been identified by Henry Holden and Stephan Zuchner's group in 2019, which is RFC1. Also, RFC1 is a repeat. And RFC1 is very likely going to be the second most common recessive ataxia following Friedreich ataxia. So what this tells us is in the recessive ataxia field, the two most common disease causes are repeat expansions. And this nicely mirrors what we know from the dominant ataxia field, namely the most frequent causes are repeat expansions. And why am I emphasizing this? This is something you don't find in the other neurodegenerative diseases. If you look at HSP, there's not even one single repeat expansion. In LS-FTD, there's one, but only one, C9orf72. In CMT, you hardly have those. So this is really something which is maybe not 100% specific to ataxia, but quite peculiar to ataxia, the high prevalence of repeat expansions. And what you also found is there's still a high share of patients with recessive ataxias which are not yet solved. And again, even larger than for the ataxia one, for the dominant ataxia ones. Here, almost half of the patients are still unsolved. Just re-emphasizing that in addition to the sequencing and the pipeline parts, which are going to listen later on, we need novel tools to tackle this upper half, which is still not yet solved. With this, I want to close my introduction. I have one question to all of you. And the question is the repeat expansions in ataxias. And I need your answer here. Repeat expansions in ataxias. What is true about them? Repeat expansion in ataxias are much rarer than, for example, in ALS, HSP, or FTD. They account for the most frequent autosomal dominant and autosomal recessive ataxias, or they have now already been all identified, so with no new repeat expansion ataxias in the last 3 years. Which of those three statements is true? And please give a vote to the poll. I don't know, Annemie, how much time do the respondents have?
Oh yeah, we will give them a little under a minute. I always strive to have more than 50% participate in the question.
Okay.
So if you still want to cast your vote, now is the time. And
again, we're only talking about repeat expansion in ataxias. So I've compared, are they much rarer? Do they account for the most ataxias? And have they already now been all identified? There were no new ones in the last 3 years.
Let's see what the audience thinks, Matthis.
Okay. Yes, that was an easy one. That was an easy starter. You were all right. They're much rarer in ALS, HSP, FTD. Nope, they are much more common. ALS, HSP, FTD. ALS, FTD just has seen an ORF. HSP has no repeat expressions at all. Yes, they have the most frequent cause for dominant and autosomal recessive ataxias. They have now already been identified. Hopefully not. We still will going to find one. And I promise you, I cannot tell you, we are going to have still big repeat expansions coming up in the next years. And in the last years, there has a nice one been identified, RFC1, as the second most common cause of recessive ataxias. With this, I want to close my introduction. I want to hand over to Andrea Nemeth. And Andrea Nemeth is going to present you the sequencing technologies.
Hi there. My name is Andrea Nemeth. I'm a professor and consultant in clinical genetics at the University of Oxford and Oxford NHS hospitals. Can you hear me okay? Excellent. Okay, so I'm going to be talking about the basics of next generation sequencing. And I'll also have some multiple choice questions at the end. Oops, that didn't work. Sorry, I'm just trying to change my slide. There we are. Okay. So the most important thing to start with understanding about next generation sequencing is the scale of genomic variation. And you will be familiar with chromosomes, which are between 50 and 350 million base pairs. And you can visualize these using what is known as a karyotype. But many genetic disorders are not visible under a light microscope like a karyotype is. They can be submicroscopic, microsomal insertions or deletions known as copy number variants, and they are detected using arrays of some description, or you will be looking at single gene conditions and individual nucleotide changes. And these are detected using sequencing technologies. That's what I'm going to focus on today. And then, of course, you've heard about the repeat disorders. CAG is a very common repeat disorder in a variety of ataxias, and they have classically been detected using PCR. But as we will mention at the end, it is possible nowadays sometimes to pick them up using whole genome sequencing. So they're very different technologies to detect a very different ranges of sizes of mutations. So Sanger sequencing was the gold standard until recently. And the major point about Sanger sequencing is that it's in series. You test gene 1, if it's negative, you move to gene 2. If it's negative, you move to gene 3. The problem is that really from the 1990s, there was a huge increase in the number of new genes that were identified, and therefore the numbers of genes that needed to be tested during diagnostics. And Sanger sequencing became too expensive to do this. So there was a big move to try and develop technologies where you could sequence a number of genes all at once, that is in parallel. And that's what next generation sequencing is, massively parallel, high throughput sequencing. So the first steps of next generation sequencing are actually very similar to Sanger sequencing. So you need to prepare your DNA, usually from a blood sample, but it could be saliva, a biopsy, or a postmortem sample. And then in the case of next generation sequencing, the DNA is fragmented, and you need to go down two pathways. One is either that you're going to enrich for the genes of interest, and that's sometimes called capture as well. And you can do this for both panels of genes or exome sequencing, as I'll show you in a minute, or there's no enrichment phase at all. You just, for practical purposes, put the entire genome into the machine. Either way, you're going to put the DNA into the machine. It's going to come out as raw signal, and then the bases need to be decoded computationally. What you will notice here is that both for targeted and whole exome enrichment, you will lose some data along the way, and that's quite important. So Sanger sequencing has a number of other variations from next generation sequencing. First of all, you get a combination of all molecules being visualized. So if you look here, you can see 3 Cs in a row in the top panel of one individual, and this individual below has a mutation. So at this first C position, only half of the reads are actually a C and the other half are a T in red. It can actually be very difficult to tell the difference between this, which is a mutation, and this, which is just a sequencing error. The other difference is that Sanger sequencing tends to be much longer, so it's around 1,000 base pairs. In next generation sequencing, you're sequencing each individual molecule, and that is a row shown along here. And the other important feature is that they're very short reads. At the moment, the vast majority of platforms use sequencing, which only gives you a couple of hundred base pairs. And so each of these molecules has got to be aligned back or mapped back to the reference genome. So what you can see here is each row is a sequence of an individual molecule that comes from the patient and mapped back to the reference genome. And what you can see here is that at this position, this G, which is in red, this person has about half of the sequence is a G and the other half is an A. So this person is variant at that position. So from a G to an A, and is heterozygous. What you can also see is here, sorry, I'm just going to move these out of the way because I can't see this bit. There we are. So you can see that's a sequence variant or an alternate sequence. What you can also see is scattered around these random bits of variation. And these are very likely to be sequence errors, which of course occur. The problem is if you only had one row, one molecule of sequence, you wouldn't be able to tell, for example, whether this was actually a real finding, as in over here, or whether it was a sequence error. And so you can tell that the numbers of times that each of these nucleotides is represented is an important proxy measure of the quality of your data. And this is known as the depth of coverage, and it's an important metric that you'll hear about probably again. The other important metric is the percentage of the reference genome that is represented. So all of these nucleotides need to be represented to get 100% breadth of coverage. And usually you should be expecting to see about 98 to 90% breadth of coverage. Otherwise, you're starting to miss a lot of those nucleotides, and that's really important. The other thing just to note quickly that I'll come back to is that if you have a repeat like CAG, CAG, you can imagine that with these short reads, it can be very difficult to map and align them back and work out where they come from in the genome. And that's one of the reasons it's so difficult. It's important to understand the difference between coding and non-coding regions of the genome. And for those of you who perhaps haven't done genetics for a long time, I'll just remind you that there are intergenic regions which are non-coding. And here is a gene with the coding exons and then the non-coding introns. The introns are spliced out to give this mature messenger RNA, and that is read by the cellular machinery to give you protein 1 coded by the exons of gene 1. The protein coding DNA is known as the exome and comprises about 1.5% of the total genome. And this means that it's very amenable to analysis and getting rid of the rest of the genome in some ways. We'll talk about in a moment. Many gene mutations are known to be located in the exome. Whether all of them are or not is a debatable point. Probably they aren't, but we know very little at the moment about the non-coding regions, so we tend to not focus on those. If we look at the different types of NGS, as I mentioned, you can have targeted sequencing. So you're literally just pulling off sequences of interest, for example, a group of ataxia genes. And so this is a phenotype-based panel. Because you're not sequencing a lot of genes, you get a very high depth of coverage, so there's good quality data. You're only going to be sequencing a few hundred genes at most, and therefore the numbers of variations from the reference are going to be relatively small, and therefore both the sequencing and analysis costs are low. And so public health systems still tend to favor this as a first off because it's a very manageable cost. However, as Matthis has mentioned, it's very limited in what it can do. And whole exome sequencing very quickly took over from targeted sequencing wherever it was affordable, essentially because it's an off-the-shelf solution. So you're still capturing all the individual exons, but you don't need to worry about what the gene is or what the phenotype is at the beginning of the experiment. You need to at the end. You get good depth of coverage, about 50 to 100 times, and you're sequencing all 20,000 coding genes. Of course, you get a lot more variance, and although sequencing costs have come down, the cost will be more than a targeted capture experiment. But as a result of this modest cost with widespread sequencing, it has been highly favored by both public and private healthcare systems, as well as for research, and has contributed to many new genes being identified. However, a major problem to be aware of is that you can get lost bases because of that capture phase where you're capturing a lot of data or a lot of genes in a very small test tube. And where in targeted sequencing, you can backfill using Sanger sequencing, there's too much to do in whole exome sequencing. As I mentioned, whole genome sequencing fundamentally is also gene and phenotype agnostic. The depth of coverage is less, but it's very, very even. And that's a very important point that I'll come to in a moment. Of course, you get vast numbers of variants because this is both the coding genes and all the rest of the non-coding regions. So although sequencing costs are still modest, the analysis costs are extremely high. And both private healthcare and research, which is well funded, tends to be the one who uses whole genome sequencing. And you must remember that you can still have some missing data. So briefly to mention about copy number variants, it is possible to identify these using whole genome sequencing. And this is just one of the concepts that has been used recently, where if I show you an exome and remind you that these are the mapped reads that are aligned back to the reference sequence, you can see that in an exome, as I mentioned, the read depth is very variable. So here it's 1, here it's 5, here it's 3, here it's 0, here it's 3 again, here it's 2. And that means that there's no way of inferring whether this is a real finding or an artifact. In contrast, if you look at genome sequencing, as I mentioned, with about 20 times coverage, if you suddenly get a region where there's very little, half or even nothing, you can suppose that there might be a deletion and look at that position in more detail. And equally, if you get double the coverage that you're expecting, then you may infer that there's a duplication. And again, look at that in more detail. So this is just one of the ways in which copy number variants can be identified from whole genome analysis.
Andrea, what was the copy number variant again? If you can just explain, or types, what are the types of copy number variants?
Copy number variants, they can be, the most common types are deletions. So you're actually losing genetic material either on one or both chromosomes. And insertions and duplications, they can be the same bit of gene that's inserted in tandem to the gene that should be there, or sometimes even they can be inserted into other parts of the genome. So you can have duplications or triplications in some rare settings. But deletions are one of the most familiar type of copy number variants seen in human disease, and they can occur in a number of different conditions, neurogenetic disorders.
Thank you.
So short tandem repeats, this includes the CAG repeats that Matthis mentioned that are the cause of SCAR 1, 2, 3, 6, 7, and 17. And also the repeats found in Friedreich's ataxia, which are intronic. Now, as I mentioned, they do not align easily to the reference genome, and it can be very difficult with short reads to miss the start or the end of the repeat. So you can't tell where it started and where it's finished. So you can't work out how many of these CAGs there might be. And when you're using short read sequencing, if the repeat is bigger than the actual length of the read, you won't be able to tell at all. In addition, if you are looking at targeted or exome capture, then it won't have intronic regions. And as you've heard, for example, with Friedreich's, this is an intronic repeat, so it's not going to be captured using this sequencing. Now, there are software programs that are being used. One of those is Expansion Hunter, but there are others. And these are very complex software programs using complex algorithms that are starting to be used in clinical practice, or at least in some research practice and in some industry practice, but they are certainly not routine. And it means that if you get whole genome sequencing, you must make sure that if you're interested in conditions known to be caused by repeats, that you check whether they're being covered or whether they need to be tested for separately using PCR. So just to finalize, there are ways of filtering this data before you get to the parts that Stephan is going to talk about that can limit the amount of time and costs, even if you do whole genome sequencing. And the general concept is that now that sequencing costs have come down a lot in the last few years, because whole genome sequencing has very even coverage, it's sometimes worthwhile actually doing whole genome sequencing, but then limiting how much analysis you actually do. So of course, the obvious place to start is an exome. So you're avoiding the limitations of exome sequencing because you're avoiding the capture. You're just going to look at the protein coding regions. But you may even want to limit this and be much more focused and look at a virtual clinical exome, which is around 5,000 or 6,000 genes, which are known already to be associated with human disease. But you may even want to be more limited than that, which is, again, even more cost effective. And these are known as virtual panels. So they're not the same as targeted capture panels. You're still doing whole genome sequencing, but only looking at a limited number of genes associated with the phenotype that you're interested in. And so this comes back very much to clinicians giving information about what the clinical phenotype is. And clinical phenotyping by clinicians is absolutely essential for this type of analysis. And this type of virtual panel analysis using whole genome sequencing is being used was being used by the 100,000 Genomes Project. It's just come out, the initial data in the New England Journal, and you can see some of the results there. And we are now using this in the UK NHS whole genome sequencing service. We don't have all genes associated yet, but we are doing both adult and childhood onset ataxias. So I think I'm going to finish there just by reminding you that different types of NGS are useful in different clinical and resource settings. They cannot identify all mutations, and therefore a negative result, even with whole genome sequencing, does not exclude a genetic cause. So, and much more research is required to find these missing mutations. So I'll finish there. I'm sure we'll have time for questions. I've just got a multiple question for you. Can you tell me which of these statements are true? Whole genome sequencing involves capturing genes of interest. Next generation sequencing visualizes the sequence of individual molecules. An exome sequencing and a clinical exome analyze different numbers of genes, and you can choose more than one in your responses.
All right, let's see if you know the answer to this question as well as to the last question. I'll give some more people the time to answer this question. Over the 50%, like I said, otherwise it's not a representative image, I think. All right, Andrea, here are the results.
Okay, so whole genome, so a lot of you got this right. Whole genome sequencing does not involve capture of genes of interest, okay? You're just taking the whole genome and putting it into the sequencer. Next generation sequencing visualizes the sequence of individual molecules. This is correct. And exome sequencing and the clinical exome analyze different numbers of genes. So exome sequencing is about 20,000 genes, and the clinical exome is about 5,000 genes. If anybody has any questions about that, please do ask them afterwards.
And I think with this, we move on from the hardware, from the different sequencing techniques. Thanks a lot, Andrea. We move on to the filtering and hand over to Stephan.
All right.
Andrea, could you share?
Okay. Then I bring my slides up. Okay, everyone. So I want to lead you through a bit of consideration of how NGS diagnostic is typically done. And I try to keep it really straightforward. As you can imagine, the challenges are pretty obvious. Number one is, as of today, we have so many different genes causing ataxia. That makes the job harder to find the one variant that is causative in one particular exome or one particular patient. The next thing is that, as you're also aware of, is that ataxia doesn't live in a silo. These phenotypes and also the genes, they overlap. And so spastic paraplegia genes, so-called spastic paraplegia genes, could also cause ataxia and so on. And there are new relationships being discovered all the time. Then you might be surprised or not, there is still a significant diagnostic gap here, meaning if you have 100 ataxia patients, how many get actually diagnosed if you apply the latest and best, such as exome sequencing, and it's definitely significantly less than 100 or even less than 90 patients. So there is a gap between what we typically diagnose when you send it off for clinical testing and the patients that are there. So more to be discovered. And finally, over the last decade, it has become clear that the genome is quite a complicated place. Andrea already showed you the different types of variants that exist in principle, but even the ones that we seem to understand best, like the single nucleotide variants, it turns out there are so many of them. There are so many of them that here in this study with 60,000 patients, or not patients, actually people from the population, they are not patients necessarily. The majority in blue here, the majority of variants that one can find in a population like this are changes that are seen only one single time in a person. And they actually have never been seen before in any other study ever. So because there's so many variants out there that are novel and super rare, it's hard to decide if a particular of those variants is actually pathogenic. So these are really some of the key challenges here when we think about which variant is pathogenic. So there are sets of criteria that have been developed for clinical laboratories specifically. Probably the most famous are the so-called ACMG criteria, American Colleges of Medical Genetics. And I have here the URL, but you can also simply Google it. It's easy to find. They come up with this quite complicated scheme here, where we consider first of all, the framework is to think of variants either as benign or as pathogenic. In reality, there is actually an in-between, which is called variants of uncertain significance. So they have neither enough support for benign nor pathogenic. Those are the tough ones, of course. And then one can be very convinced that something is benign or pathogenic, very strong, or not so much convinced. So there's a certain grading here. And what goes into this framework are different sets of considerations, such as population data, how common are certain variants, computational predictions, and so on and so forth, functional data. And not all of this might be available for a given variant. In fact, for many variants, it might be quite spotty, the evidence. So that makes the whole enterprise a little difficult to conclude often. And also, once this is done, you may notice here in this complicated table in the background, there are these, PS1, PVS1, these abbreviations. Once this is sort of all considered, you can come to a final consideration, which goes from a grading from pathogenic through likely pathogenic, benign, likely benign, uncertain. That's how clinical genetics works today. But to give you some more sort of real hands-on idea here, again, as you just heard, what is in an exome? There are about 20,000 coding variants to be expected in any person's exome, really. With several hundred of those have never seen before, neither in control databases nor in other patients. And then, of course, don't forget, there are false positive calls, always. And there are, of course, false negative calls. There are variants that we simply miss for often technical reasons, really. So, and the goal is, of course, in any given exome, to find the one variant that can explain the patient's phenotype. So, here are some really straightforward considerations. Number one is obviously inheritance pattern. If you see a patient and you can work out the family history, and you actually get clues that find other relatives that indeed support a dominant or recessive inheritance, that already is very, very valuable. Sometimes there's consanguinity that can also be helpful. At least it could be a guideline. So taking family history is very important. Another one is the quality of variation. So you sit there, it's your task to work out which of these many variants in an exome is the real one. So, but just adding, trying to reduce the false positives and is a big help. So typically this is applied in the filtering settings. And one of the biggest really is considering the allele frequency. That's why I put this in red here. This is a fairly simple but important criterion. We are talking here in ataxias, of course, about rare diseases with a low prevalence in the population. So we should really only consider variants as potentially pathogenic that are also quite rare in the population, right? If a variant is common in the population, like say 20% of people, then ataxia should be more common, right? So we really look for quite rare variants. And here are some examples for how rare they should be. In recessive cases, we would typically look for variants that are in about 1% of controls, okay, but not more necessarily. And for dominant cases, we really, in the typical case, we don't want to see them in controls at all. But assuming they are fully penetrant, right, controls should actually get ataxias. Now, in these databases, of course, there might be a single case, sometimes two. And then it's, difficult to make a decision, but there are certainly examples where true pathogenic variants are in these databases sometimes. So how does this look? Very practical. One of the tools we use in the ataxia world to do research is this Genesis tool, and these are just sort of snapshots, and I took a specific case here. This is a case of a recessive ataxia family. So we know it's recessive. We know the phenotype. That's great. And I just apply these 4 filters that I showed you. And I actually only apply the quality filter to begin with. I don't want to get all these super low covered, low quality variants. If I do this and I do not consider the other ones, I get over 10,000 records back for this one case. Okay, so that obviously is not super effective to just do quality. Now, if I add to the quality filter the allele frequency, so I would only consider variants that are seen in less than 1% of the population, I bring these 10,000 down to about 500 records, 500 variants. And when I then add to this the inheritance pattern, it's recessive. In this case, we know it's recessive. It drops to 68. So now from 10,000 to 68. Okay. Now, and importantly, we know what the diagnosis is. For clinical testing, we would really focus on known ataxia genes. This is not a research study. In a research study, we are open-minded. Clinical diagnostic labs, they have to be more adherent to what is reasonable. So if we only consider ataxia genes, it actually drops to 4 records. And when you inspect those more closely, you see that there are 2 genes here, ATM and STUB1. And so basically it tells us these are compound heterozygote changes, 2 in each gene. That's why they come up. So we drop to 2 genes here, 2 possible solutions by applying these 4 filters from 10,000 to 2. Okay. This is probably a somewhat fortunate situation, but you can see how powerful these filters are. And in this particular case, we know that STUB1, actually these 2 here, are the ones that turned out to be the causative gene. So when I show you this live, this is the same, what you just see from the screenshot. This is actually in the database. So I go to STUB1 here, for instance, and this is just a feature of the software. You can see the criteria here on the left bar. And there are other criteria, of course, that you could take into account, and professional geneticists do this. One of them is functional predictions. You can see there's a whole list of different computer algorithms that can guide you, and they already tell you that this particular computer algorithm thinks this is damaging, or this one thinks it's disease causing, right? So this is taken into account as well. The other things like gene or base constraint that people take into account, which transcript is it on, and on and on. So splice predictions, for instance. But I don't want to overwhelm you with this. I think going with these 4 basic filtering already, you can do a great job. So that's pretty much how this works. And I don't know if you take questions now, otherwise I will move to the Q&A, I have 2 questions.
There are not any questions from the audience yet, but I do invite you to write them in the Q&A box or in the chat. And we will first do Stephan's multiple choice question while your questions come in.
And I hope I'm not too fast here. Okay, so the first one here.
Do you want to read it out, Stephan?
Sure. So what are some of the key challenges in identifying causative variant in an exome? A, the amount of rare variation in any exome, the locus heterogeneity of ataxia, and C is functional data confirming pathogenicity of variants is limited. Or D, all of the above.
What does locus heterogeneity mean?
Oh my God. It means that you have multiple genes, different loci.
So you have many genes which might cause ataxia.
Linked to the same phenotype, like ataxia. Basically, many genes can cause ataxia. Instead of genes, we like to think of locus, a genomic locus. Yeah, I should have explained that.
Well, you get the chance to do so now. So the question for the audience is this one of the key challenges in identifying the causative variants? Well, I'm at 52%, so I think that's good enough. Do you want to take us through the results, Stephan?
Fantastic. You got it all right. That's great. As I said, the amount of rare variation really is one of the key challenges, I'd say, in figuring out which single one or 2, in this case, recessive are the pathogenic ones. Often patients carry mutations that have never been seen before, for instance. It's just, that's called allelic heterogeneity, by the way. Then the locus heterogeneity is a big problem. It's kind of linked to that. The more genes we look at, it's sometimes more difficult to work out which of the ones is correct. In this example, I showed you 2 genes as potential solutions. So we didn't go into this, but obviously we made a decision it's step 1 and not ATM. And then C is functional data confirming pathogenicity of variants is limited. That is true. If we had more functional data, it would probably be easier to convince us which one is pathogenic and which one is benign. So all of them are important. Mm-hmm.
Stephan, in the meantime, a question came in for you. I think, can you identify cis-trans variants on next generation sequencing?
Excellent question. Yes and no. Typically no. What we use today, the NGS, next generation sequencing, is mainly a technology that produces short reads. Okay, so Andrea showed you there are these horizontal bars. These are all reads, and we try to cover the target region with many reads, but each read is quite short, about 150 base pairs. So you cover only a portion of the exome, and so one read might support a variant at a particular place, but if you have a compound heterozygous change on another exome, that particular read wouldn't reach that far. So the read on the other exome is not really linked in any way to the first read. So that's why you can't really work out just based on that if it's in cis or in trans. Sometimes, by happenstance, the 2 compound heterozygous changes are very close to each other and they fall on one read. If that happens, you can actually work out if it's cis or trans. So in the future, we think the field will eventually have long read sequencing available. I could imagine that then you could actually have very few reads that span the entire gene, and then you could actually see that. I hope that answered the question.
Thank you, Stephan. I think we will go ahead. Yes.
Yeah. If you want to one more question, you like that? Or
yeah, let's.
So that's the other way around. Which statement is wrong? There's no standardized framework for variant interpretation. Math, minor allele frequency, is an important consideration in variant interpretation. C, machine learning techniques may become a part of variant interpretation. We haven't talked about this. We'll talk about it later. I hope you can guess the answer. And D is exome sequencing cannot detect all genetic changes in a person's exome.
So the question is, which one is wrong?
Correct.
Yes. Some more difference in opinions here.
Okay. Let's see.
A few more answers from the audience. We like to know what you think. I really have this thing for the 50% mark, guys. Sorry, that's just a quirk. So 2 more people need to answer. Yes, thank you. Here we go.
Yeah. So I showed you there is the most commonly used framework, though not the only one, is the ACMG framework, and so that exists actually. The other ones are, they are indeed true. So minor allele frequency in the population is important to consider. Machine learning, you can imagine automated techniques are being explored and will only become more important. And now D might be somewhat misleading here. What I really meant is, each single exome certainly might have places that it doesn't cover well, for instance, and that is true. That not all the single exons are sometimes sufficiently covered, or there might be exons we don't know about. They're not covered by design, for instance. So there are certainly shortcomings of the technology. I guess the point is, I want to make sure you understand, this is not when we say whole exome sequencing, it really is not the entire, it's not all variants in exomes. You will miss some. It's just how life works. It's not a complete set. It might be quite high, 95, 98, 99% even, but you will always miss some. And sometimes this is the one you're looking for.
Okay, thank you, Stephan. So I think with this, we close our session for now. Just leave the last 2 minutes for discussion. And we will not get to the science part. We get lots of pipelines. We can do this in another webinar to come. But we will just, I think, just to highlight the next steps in Ataxia genomics is that we really try to tackle those parts of the pie which are not yet solved. And for this, we will bring all our exomes and genomes together. And if you have larger exome or genome data sets, those can be uploaded, for example, in the Genesis pipeline, which Stephan has shown you, and thus become part of a collaborative pipeline, and you can work on them. So if you have data sets which you would like to work upon, share in a collaborative fashion, please contact us. Or if other genetics questions, we're happy to answer them, in particular in the Ataxia field. But with this, I would like to invite you. I think we have 2 more minutes, maybe, Anne-Marie, for further questions. And we're happy with anything. Can be technically, can be related in the Ataxia gene, whatever you would like to know about the Ataxia genetics.
I think there was one question I falsely put into the answered section. Can you please talk a little bit about pseudo gene problems in NGS from Noemi? Yes, that's one example where we know, because of certain findings recently, where the technology can be misleading, that the short read sequences may not be able to differentiate between a real gene or what we call a pseudogene. Pseudogene is a copy of a gene, and they exist. It's an evolutionary copy that is not functional, but could be so similar that the short read sequences cannot say this is the real gene, this is the pseudogene. And that's another way where one might miss an existing variant.
Are there any other questions? You can also write them in the chat. I will see them there as well. Maybe a comment from one of the panelists still?
Sorry, didn't want to interrupt you. Feel free to add more questions, of course, but maybe a question from the more diagnostic point of view. So if you are in a diagnostic setting and you want to answer if there's any gene related to the clinical phenotype, maybe 2 related questions. What do you think is more important, the information of the family history, that taking into account that it can be misleading or incomplete, or do you think the clinical phenotype is more important? And regarding the clinical phenotype, what do you think is better, to have just the one leading symptom like ataxia and nothing more, or as concrete and as complex description of every sign and symptom that is presented?
Matthis, you want to take that one?
Stephan, you go for that one.
Who needs to answer?
Maybe I can start off with first impression, then the others can jump in, because I don't think there's not a 0 or 1 answer to your question. Yes. So I think the mode of inheritance is really one important filter Stephan has shown. It's really just, if you look for a dominant versus a recessive gene, that's just so important. So I think that is a strong filter. On the other hand, for the phenotype, I think what is already, at least the basic phenotype should be right. So it should be, are we talking about ALS or ataxia? And of course there's overlap, but I think the basic phenotype is right. And from this, I would say both is already a good starting point. Then, of course, the more phenotypic information you have, the better, of course. But at least if you got the mode of intelligence right, and if you get the phenotype right, those two are both nice filters. Because for them, you can filter for the mode of intelligence, and for the phenotype, you can already know I have to focus on ataxia or on HSP or ataxia HSP phenotype. So I think both basic information is right, and I would rather have those two right than having the 10th phenotypic aspect. This would not help me as opposed to those two basics. Stephan, what do you think?
Yeah, I agree. There's probably no right or wrong here necessarily, but I would also say that, you know, sometimes when you submit when you order a clinical test, genetic test, and all you give the clinical lab is ataxia, right? That may make it a little hard for them sometimes. It's better to give them a bit more detail. I give an example. The two candidates in the example I showed you were ATM and STAB1. Now ATM actually codes for, I mean, it causes ataxia telangiectasia. And so those patients look a little different, right? If you just say ataxia, makes it a little harder sometimes for the interpreter.
The other thing I would add is that, of course, a number of neurodevelopmental disorders that include ataxia are de novo, and so there isn't going to be a family history. So we often, I'm a geneticist, so of course I'm very keen on getting the family history data whenever possible. But if family history data isn't possible, then it can still be, or if there isn't a family history, then it can still be very important to get all of the patients sequenced, the parents and the child, and identifying a de novo mutation is an important part of the ACMG guidelines.
And so, Jenny, I think maybe to sum this up, I think maybe 2 or 3 phenotypic features would be good, but also the age of onset is very, very important. In addition to the phenotype, are we talking about like developmental disease, or is it starting age 60? It's a big time filter also for this. So that's important if you order a test to add this.
And keep in mind these professional clinical geneticists who interpret these exomes, they are trained to understand phenotypes. They maybe not be trained to examine patients, but they do understand. That's part of their training, to really understand complex clinical phenotypes. So it's certainly worthwhile to give them a bit more information.
Okay, then I would maybe have a very short last question to the last point, Stephan, you mentioned, and Matthis, you also somehow mentioned it. So what would be your idea? What is the best infrastructure that or framework that could be built up to support the identification of new genes in general? I think you answered already with the idea of big databases, but in particular, how do you think that AI, so artificial intelligence, will help, or where do you see the potential for AI techniques? Where exactly can they support? Should they just be trained to better identify already known variants, or do you see more the potential in really the new development and the new identification by searching in this big data for patterns that have not been identified before? Sorry, or is there a third option?
Yeah, no, it's a good question. I would say we are currently in a phase where a number of groups explore the possibility to automate variant interpretation. Right, there are sort of more hard-coded ways. You know, this minor frequent. So the ACMG is a good example for almost a hard-coded way of looking at things. And then the question is, could there be something that's, quote, a little bit more intelligent, like a chess like a chess computer program that actually beats a human, right? How does it do that? And the judges are still out there. You could argue both ways. For clinical diagnostic, you really want to be sure this is correct. So maybe it is a good place, maybe it's a dangerous place to start with. In research, we're allowed to do, on this front, we allowed to do what we want. So we definitely see already some utility in the research space. But the judges are still out there, how much better these things will become.
But I think, Jenny, what is important is, so this ML-based will be a process, maybe some tools in the future, but I think what we can already do now is actually the first part of the question, or the first part you gave yourself, is a big time problem we are having is that the data sets are still in silos, and mostly the data sets are in diagnostic silos. We send our exome genome somewhere, then somebody looks at them, maybe once, maybe twice, a few years later, and then it's pretty much stopped there. And the bugs, and then they either solve or not solve, but then the diagnostic job is done. But we don't have very often a solution for our patient. So what we need to do is we need to get the data sets out of our diagnostic and research silos and bring them together. And that by re-examining together over and over and over and over and looking at them again in large and larger aggregations, this is actually the main next step to find new genes. And the pipeline Stephan has shown you, Genesis, this is there being done, and this is why there have been 17 ataxia genes found just in the last 5 to 6 years, and the number is increasing. This just shows the power of bringing those data sets together. So I think rather than for all of us not being primarily geneticists, I think the next step is really try, and this is something, given that the diagnostic data sets belong to the patient, at least that's in Germany, they're paid by the healthcare, and the patient can decide where his diagnostic data set has to go. They're not they do not belong to the industry, they belong to the patient. So this means if the patient justifies you to use his or her diagnostic data set, you can then also bring them together in a joint aggregation of data sets. And by bringing them together, that's the main tool of reanalysis and thus finding the genetic cause. I think that's a major next step we should focus upon.
Yeah, I think it's unquestionable the necessary prerequisite. So without, no, I can be trained. So you need this large samples. Okay, then.
All right. In the meantime, no more questions from the audience came in, but I think your final discussion really added something to this webinar.